Okay, welcome back everyone. At this time, I would like to introduce Dr. Jeremiah Johnson, Associate Professor within the Department of Microbiology at the University of Tennessee. Dr. Johnson's presentation is titled Identification of Genetic Determinants that Facilitate Campylobacter jejuni Survival During Poultry Processing.
All right, thanks Denise. Happy to see there's a commuted few that stayed behind. So we're going to actually take a little bit of a detour, I think, from most of what you guys have been seeing so far today. As opposed to somebody who might come from a poultry science department or exclusively an ag department, I'm actually more of a molecular microbiologist that focuses on human medicine. One of the things that's particularly relevant within our research is that we use a lot of molecular tools to sort of examine the ability of a significant bacterial pathogen in host colonization. So one of the things that I really like to start people with in my talks is to remind them that diarrheal diseases are actually still the leading global cause of morbidity and mortality. So if you take all of the different types of gastrointestinal infections that happen every year on this planet, it's about 4.5 billion episodes of diarrhea, and a lot of those are going to be due to infection. And just because of the scope of diarrheal disease globally, it's also a pretty significant problem when it comes to loss of life. So it's about number 8 globally in terms of sort of human killers, we'll say, with anywhere from 1.2 to about 2.5 million fatalities caused every year. And really one of the unfortunate things when it comes to these diarrheal diseases is that low and middle income countries, not places like the United States, tend to be those that are most impacted. So in places that have typical clean water and sanitation infrastructure and things like that, we're able to avoid most of these types of causes of infection. But obviously people elsewhere in the world without those resources are not able to avoid these types of infections. And so there's a lot of viral causes of gastrointestinal disease globally, for sure. So if there's any virologists present, I don't want them to think that I'm hating on them. But when you start looking at bacterial pathogens, you'll notice a lot of sort of the who's who of bacterial gastrointestinal pathogens, including enterotoxigenic E. coli, non-typhoidal Salmonella species, and Campylobacter species. And so, really what my group focuses on predominantly is really trying to understand from the bacteria's perspective, what does it possess that allows it to colonize different animal hosts, transmit from those animal hosts, and actually infect and cause disease within humans. And so one of the things that's becoming really apparent within the Campylobacter field is that there's really a bifurcated impact on human health. So when we typically talk about high resource settings like the United States, where it's still one of the leading causes of bacterial-derived gastroenteritis, we see a lot of quite a different impact on the host. And so typically what we would experience from a relatively low inoculum is really intense gastrointestinal distress, high fever, abdominal cramps, bloody stool. And though you may want to die for a day, most of the time this is actually more of a self-limiting infection. So most of us would tend to recover really without incident, just typically convalescing at home. But one of the big problems is that Campylobacter is actually emerging as a really significant pediatric pathogen. So globally, children under 5 are actually one of the most hardest hit. So if you look at these original numbers here in terms of 4.5 billion episodes and about 1.5 million deaths. More than half of all of those are actually in pediatric patients under 5 years old. So them oftentimes acquiring these bacterial pathogens from, oftentimes contaminated water sources, and even beyond just sort of the more drastic acute types of infections, there are also, sort of more subtle persistent infections that occur. So Gates Foundation is actually dedicating a lot of resources to try to understand how Campylobacter species are leading to the development of what's called environmental enteric dysfunction within these children. Because one of the things that emerges within these kids is that they can no longer acquire nutrients as readily, and their growth is severely delayed. So with all this said, there's not only really an interest in agricultural health in terms of inhibiting Campylobacter infections, but also in global health as well. And so really one of the reasons for just the predominance of Campylobacter infections is that the organism asymptomatically resides within a lot of different types of livestock. It's really historically been associated with poultry. So it really likes avian hosts, gets to really high gastrointestinal loads within the cecum of chickens. And then, of course, either direct transmission from the feces or contamination of poultry products and then undercooking in the consumer's home. The person can then actually acquire the Campylobacter from relatively low infectious doses, several orders of magnitude. So, millions and millions times more infectious than, say, E. coli or Salmonella species within humans. One of the other things that actually my group has been working on as well is that we're also finding that cattle are a pretty significant source of Campylobacter infection in humans as well. So almost up to 50-60% of cattle that we survey, they also have Campylobacter within their gastrointestinal tracts. And so really from all of these sources, you can imagine that there's going to be a lot of environmental contamination with Campylobacter that then is going to naturally wind up in surface waters. And there's actually a lot of clinical case studies out there where people just, participating in normal recreational water activities wind up with Campylobacter infection. And so because of this, the association with the Campylobacter in these different animal species, there's a lot of work, both at USDA and FDA, as well as in collaboration between us and USPOULTRY, to really try to figure out ways to eliminate either loads within these animals or the ability of the bacterium to transmit from the animals to people. And one sort of set of concerning conclusions that I just wanted to communicate to you guys is, and I didn't have time to share this data today, but I kind of alluded to it with the pediatric impacts, is that Campylobacter infection isn't really a simple acute gastroenteritis anymore. There's a lot of inflammatory diseases that are becoming increasingly associated with Campylobacter infection, different types of arthritises, even blanking on the term, brain types of inflammation and things like that. And the other thing that I wanted to really drive home is that a lot of the previous work that's been done in this field, the rates of Campylobacter infection aren't going down. So all of the work that's been done up to this point really isn't impacting transmission of Campylobacter to humans as far as we can tell. It's still relatively high rates of infection. And so really what my group specializes in as microbiologists, molecular microbiologists, are using different genetic tools, as I was saying earlier, to try to address what features does the bacteria have that allows it to colonize within these hosts and then survive transmission from the host into humans, and then once it gets into humans, what does the bacteria possess that allows it to actually cause disease? And so the one thing that I really want to kind of communicate to you guys is that realize that a bacterium is a living organism. It's not a virus. These have their own metabolism. They have their own preferred states that they want to be in. Campylobacter, despite its ability to cause all these infections, is actually kind of a finicky. I sometimes joke with my kids that they seem to be have as many preferences as my children do. They like certain temperatures, they like certain atmospheres. And so because of that sort of finickiness, one of the things that we can sort of leverage is our genetic know-how to create different mutants of Campylobacter, and then essentially ask, if we get rid of gene X or gene Y or gene Z, do you lose the ability to actually persist within a host, persist during poultry processing, persist during storage, or even colonize or cause disease within a human host? Okay. And so to, of course, get at all of those questions, we developed this transposon mutant library. So transposons are essentially little pieces of DNA that can hop into a chromosome and interrupt those genes that I was talking about, so either gene X, gene Y, or gene Z. And so we actually created this library with past USDA funding where we have about 8,000 individual mutants. And so what you'll see here in this picture is these are 90 plates that have 96 wells in them. And so imagine the tortured grad student that had to do this. There are 90 plates with 96 wells, and each one of those has an independent mutant within it. So a gene X mutant in one, a gene Y mutant in another, a gene Z mutant in another. And so by surveying those, we can actually sequence that entire library. And from that, we find that, there is a little bit of redundancy. So there are sort of twins, so to speak, within the library. But importantly, there's more than 6,000 individual mutants across all of those plates. And through those 6,000 individual mutants, we've been able to inactivate about 60% of the 1,700 genes that Campylobacter has. And so what we were simply able to do with that is using a 96-pin replicator. You can stamp these out onto a growth media. You can put them into the environment Campylobacter wants. You can grow them. And you can see here in A the result of that, where we have 9 Campylobacter colonies that grew. But then at the same time, we can also move a duplicate of that plate to what we would consider a non-permissive environment, like storage conditions. Let it sit for a few days. So we let them sit for 72 hours just in a refrigerator, and then move them into the permissive environment, and then let each of those individual strains grow up. And what you can see is that in this box here, we had one strain that was viable coming out of the library, but after it sat in the refrigerator for 3 days, it died. So it wasn't able to be recovered relative to these other 8 strains. And so after doing this entire screen, again, back to tortured graduate students, we found about 160 examples of this where there was a growth observed, but not after it was stored for 3 days in the refrigerator. And so the next thing we did was actually we wanted to quantify this some. So, this is pretty, colony, no colony, pretty binary, not very quantitative. And so what we did, back again to tortured grad students, was they took all of those 160 mutants, created an inoculum of those, plated those on normal Petri dishes. Similar to this before, we took one, put it under permissive conditions, and then took a duplicate set and put it under non-permissive conditions. And then after 72 hours in the refrigerator, moved it back to permissive conditions, and essentially see how many of the individuals that were originally plated how many came out, how many survived. And so again, bacteria are clones of each other. So all of these individuals, these are all of mutants of gene X, for example. But you can get some quantification of how susceptible is gene X to cold storage by how many of its clones die over that time. And so we were able to quantify all of these. So you can see here, this is normal Campylobacter. Campylobacter, like I was saying, doesn't really prefer this environment. So you can see that about 6.5% of Campylobacter remains after just being stored normally. But all of these are different genes that have been inactivated. And you can see that we get decent reductions in survivability, some of them more than others. Some of these you can see that it's just fractions of a percent. And so one of the things that was done several years ago was that some mathematical modelers had actually made the prediction that if you can reduce the loads of Campylobacter in chickens by just 2 orders of magnitude, you can actually reduce human infections by about 30-fold. So you're basically driving it from that 1.3 million cases to just a few tens of thousands of cases annually. So being able to go from about 6.5% survival during storage to under 1% survival could have an enormous impact, of course, on human health. And again, USPOULTRY helped us fund all of this research. And so from this, we basically prioritized those mutants that had the strongest survival defect. So again, tortured graduate students. We go from that 160 down to about 60 mutants that we prioritized based on how extreme the survival defect was, where these mutants are only surviving about 0.3% to several decimal places to the right compared to the normal parental Campylobacter strain. And so what we did with these is that, from a geneticist perspective, several of the organisms, like I was saying, were made with this transposon that hops into genes and inactivates it. But we actually have the ability to use genetic engineering to create what are called seamless gene deletions. So we can actually remove all of these, gene X, we can remove it seamlessly from the genome as if it never existed. And so we did that for all 60 of these different genes that had these strong survival defects, and then confirmed whether or not that survival defect remained. And so from doing that, one of the things we found is that for 7 of these gene deletion mutants, the survival defect did remain. So we did lose quite a few of those. Something had happened with that, and again, who knows with genetics. Something had happened with that earlier strain that allowed it to have sort of that survival defect. So we confirmed it for 7, and then what we did with those was, at this point, we had been using Petri dishes with normal bacterial growth media, but we wanted to actually look at survival on actual chicken meat. And so what one of my students did was take 1 centimeter cube chicken pieces from a local grocer and essentially dipped these chicken cubes in all of these 7 mutants, or individually, I should say, within these 7 mutants, and then stored them for up to 7 days under storage conditions, so sort of sealed and on absorbent paper, just like you would at the grocery store. And then over time, sort of track how survival of that mutant, whether survival was decreased over time over that 7 days. And so one of the things I'm showing you here is just at the end of the experiment. So at the end of 7 days, you can see here, this is the normal parental strain that we just call essentially 100%. And then the survival of those 7 strains relative to it. And what I want to really point out here is that in this particular gene, just called 1304, we essentially get no recovered viable bacteria after storing it on chicken meat for up to 7 days. And so then the last thing that USPOULTRY helped us do with this project was we wanted to see how this mutant, we know that it's completely defective for storage on chicken meat, but we also want to see how it works actually in colonization of chickens. And so what we find here is that if we take the normal parental strain, we do by oral administration to little day-of-hatch chicks. We let that sort of establish for 7 days, and then we harvest the cecal contents from those birds. You can see that in those that were colonized with normal Campylobacter, we have really high loads, about 10 to the 7. So, you're talking about, 10 to 100 million cells per gram of content of these, cecal content of these birds. But when you look at that 1304 clean deletion mutant, you can see that we drop that down several orders of magnitude. So not only is this gene required for survival of this bacterium on chicken meat, but it's also required for actual colonization of the bird. And so in closing, one of the things that I kind of want to leave you with is that, future research could be done on this, this gene. It's actually predicted to encode for an enzyme that allows the bacterium to, if you're not into sort of central carbon metabolism, which who is, the gene is required for the direct conversion of malate to pyruvate, which is really a derivative of sugar sources that gets directed into energy production in the citric acid cycle. So we could potentially come up with inhibitors of this enzyme that would apparently not only inhibit colonization of Campylobacter within birds, but if you could make a sort of irreversible inhibitor of this enzyme, even if it were able to colonize, even if it got out of the bird, it would have we would predict that it would more readily die on the poultry products as a result of that. And so with that said, this is my beautiful group at University of Tennessee, all the funding sources, and of course USPOULTRY. Thank you for the support.
I do have a question. So, will your laboratory
It's the first one you've asked.
I know.
Come
I was waiting
on.
this
Well,
one.
I feel singled out.
Sorry. You're
It
special.
makes me special. Okay, yeah, see, there we go.
Will your laboratory be continuing this work, or have you already begun continuing this work? Because this
Yeah.
is something that could be groundbreaking for the poultry industry, as we have so many difficulties addressing Campylobacter. As you know, a lot of work goes on with Salmonella, but unfortunately not as much occurs with Campylobacter. So
Yeah, I always joke that I wish we had Salmonella's publicist.
yes.
So we would like to, for sure. One of the things to do now, of course, is as molecular biologists, we actually have the ability to clone and express this enzyme. We can purify it, and there's actually a lot of really simple, cheap, I should say, reagents that would allow you to find molecules that inhibit the activity of the enzyme. But everything requires money, so we need money to do that. And so one of the hopes is, unless you want to,
Let's talk about it.
yeah, let's talk about it, or submit something to the USDA and see if they would be interested in funding it.
Thank you.
Thank
Great
you.
work. Any
All right,
questions
appreciate
any additional questions for Dr. Johnson?
Come on, Denise asked one. No, nobody.
All
Okay.
right.
Thank
you
you.
so much.
Appreciate it.